Hi everyone and welcome again. Let us continue our talk about nucleic acids. In the last video, we focused on DNA, its structure, functions, and the process of DNA replication, which is essential to our life and the survival of the species. Today we will talk about RNA, the other main type of nucleic acids. And we shall see how the cooperation between DNA molecules and RNA is essential for life. RNA chemically is similar to DNA. It is also a chain of nucleotides connected by covalent bonds. However, there are some major differences between DNA and RNA. First, the sugar in RNA is ribose sugar, not the deoxyribose sugar present in DNA. Second, RNA is a single-stranded molecule, whereas DNA is double-stranded, as we have seen. Third, RNA contains nitrogenous bases adenine, guanine, cytosine, but contains the base uracil instead of thymine, which is present in DNA. Furthermore, there are three main types of RNA molecules: messenger RNA, transfer RNA, and ribosomal RNA, which have essential functions in our life. Let us have a look at their general function briefly. Messenger RNA carries and transfers the genetic information from the DNA residing in the nucleus to the cytoplasm, the area outside the nucleus, in order to be executed and use the genetic information in making proteins. Transfer RNA carry the amino acids, which are the building blocks of proteins, and transfer them to the proper site for protein synthesis. Ribosomal RNA plays role in the structures of ribosomes, which are the factories specialized in making proteins. We mentioned before that DNA has the genetic information needed to define all traits of the living organism. But how the genetic information is transferred and expressed as traits of the organism? Now it's the time to talk about genes and how they are expressed as traits. Let us look at the concept of the gene. The nucleotide units along the DNA are arranged to give a specific sequence that has a specific meaning, somewhat similar to the arrangements of letters in words and phrases to give specific meanings. DNA, as the book of life, contains the instructions written by a specific type of language, the genetic language, which is manifested in the genes. The gene is a sequence of DNA containing the genetic information needed to make a specific protein, or to make an RNA molecule, which ultimately results in certain traits. So genes are the DNA units responsible for genetic traits. Each gene may be responsible for one or more traits. Traits such as skin color, body dimension, any external or internal traits in the body. It is very important to know that the expression of the genetic instructions and information into traits is a very complex and at the same time fascinating process, which involves the cooperation of many molecules and structures in the cell. The following is a simplification of the many steps performed to convert the genetic information in DNA into genetic traits such as the color of the skin. The first step in gene expression starts when the genetic information in the gene is read and copied in the form of messenger RNA molecule. This process is called the transcription. To rephrase it, transcription is making a copy of the nucleotide sequence of the gene. This copy is called messenger RNA. Now, messenger RNA molecule contains the same genetic information which is present in the corresponding gene, and the gene must stay safe, protected in DNA in the nucleus. But messenger RNA has the freedom to go out of the nucleus to the cytoplasm to deliver the information to the protein synthesis factories, the ribosomes. Now we come to the second major step. The ribosomes then read the information in messenger RNA and translate it into making a specific protein, a process called translation. To build proteins, the transfer RNA molecules carry the amino acids, the building blocks of protein, and place them at the proper location in the ribosomes, the protein factories. The ribosomes, in turn, link amino acids next to each other to make the protein chain, which is converted to the final protein molecule later. The conversion of the genetic information in RNA to protein synthesis is called the translation process. So, the expression of the gene involves two main processes, transcription and translation. However, there are some RNAs that are never translated into proteins, and these have important functions too, such as transfer RNA and ribosomal RNA. Another question popping to the mind: how are the nucleotide units in messenger RNA converted to the protein units called amino acids? The answer is in the genetic code. So what is the genetic code? The genetic code is a set of rules used by living cells to translate the information encoded within the genetic material into proteins. It is a triplet code having 64 possibilities. In this code, a sequence of 3 nucleotides called a codon in the messenger RNA molecule specifies particular amino acid in the corresponding protein molecule. The code is written as RNA with a uracil U instead of T because it's RNA that's being translated. Now, I guess we need an example. Let us take some. The codon GCU in messenger RNA specifies the amino acid alanine in protein. The codon CGA in messenger RNA specifies the amino acid arginine, and so on. Furthermore, there are codons for the start and end of protein synthesis to define the length and type of the protein molecule. AUG is a common initiation codon indicating the start site of protein synthesis, which means the first amino acid in the protein chain. And the codons UGA, UAA, UAG are stop codon indicating the end of protein synthesis. So, a gene that codes for a protein has a start codon followed by a series of codons that encode amino acid sequence in the protein molecule, and at the end, a stop codon. A gene is more than just the sequence that encodes the protein. It also includes sequences involved in the regulation of gene expression, such as promoter sequences that define where transcription starts and are the sites where the proteins involved in transcription binds to the DNA. Let us take an example of how genes determine the trait. The skin color trait is determined by the amount of melanin protein produced in specialized skin cells. Differences in the amount of melanin produced give rise to the variation in human skin color. The more melanin amount, the darker the skin. The amazing thing is, there are many genes and factors interacting in complex ways to determine the amount of melanin produced. So many genes produce many proteins which interact to regulate and determine the amount of melanin produced in the body, which in turn determines the skin color. Generally, the same principle applies to other traits. So it is the cooperation between DNA and RNA is what makes us the way we are, the way we look, and the way we behave. And this is another example of the symphony of life being played all the time in the unseen microscopic part of our bodies. Thank you for watching, and see you in next videos to continue our tour into the amazing world of the molecules of life. Your questions and comments are always welcome. Goodbye, and see you later.